Welcome back to another Paper 2 video of the Paper 22 series. This is the May-June 2015 Paper 22. If you are new to this channel, you know what to do. Like, comment, subscribe, and finally, watch and learn. Without wasting further more time, let's get into it. Question 1. Each of the statements A to E describe a structure associated with the mammalian heart. For each statement, identify the structure that is being described. A. The chamber that pumps blood into the pulmonary artery. The keyword over here is chamber, and it pumps blood into the pulmonary artery. Where is the pulmonary artery situated? It is situated towards the right side of the heart. Therefore, it should be the right ventricle because the blood flows from the right atrium into the right ventricle and then from the right ventricle into the pulmonary artery. And since the question refers to the chamber that pumps blood into the pulmonary artery, the right side has two chambers and the chamber that pumps into the pulmonary artery is the right ventricle. Statement B. A blood vessel that transports deoxygenated blood into the right atrium. This question right here refers to the blood vessel, not a chamber. And the blood vessel that carries deoxygenated blood into the right atrium is nothing but the vena cava. Statement C. The specialized tissue responsible for delaying the conduction of impulses from the atria to the ventricles. This right here refers to the cardiac cycle, where there is a mention of the atrioventricular node. The atrioventricular node causes a delay when sending excitation waves to the ventricles. Therefore, since the question refers to the tissue that delays the conduction of impulses, it should be the atrioventricular node or the AVN. Statement D. The blood vessels that supply cardiac muscle with oxygenated blood. This one, of course, should be the coronary arteries, as mostly the arteries carry the oxygenated blood, except the pulmonary artery when referring to the mammalian heart. Therefore, the blood vessels that supply cardiac muscle are the coronary arteries. Statement E. The valve that prevents the backflow of blood from the ventricle that contains oxygenated blood. Valve present on the right side of the heart is known as a tricuspid valve, and the valve on the left side is known as a bicuspid valve. And as you know, the left side of the heart brings the oxygenated blood. Therefore, since the question refers to the ventricle that contains oxygenated blood, it refers to the left side of the heart. And what valve is present on the left side of the heart? It is the bicuspid valve. Hence the answer is bicuspid valve. And this question refers to the valve that separates the atrium and the ventricle, because preventing the backflow of blood from the ventricle is the atrioventricular valves. Preventing the backflow of the blood from the aorta or the pulmonary artery would be the semilunar valves. But this question refers to the atrioventricular valve that is present on the left side of the heart. And the name of that special valve is the bicuspid valve. Question 2. Figure 2.1 is a scanning electron micrograph of an area of the trachea showing the presence of B. pertussis bacteria. B. pertussis is the causative organism of respiratory disease in humans known as whooping cough. The disease is transmitted from person to person in a similar way to tuberculosis. A symptom that is common to TB and to whooping cough is the production of an excess of mucus. Part A. Describe the damage caused by B. pertussis that is shown in the area labeled X on figure 2.1 and explain how this will affect the functioning of the epithelial tissue of the trachea. If you look at this specific diagram given and at the area labeled X, what you would see are the cilia, and you could mention the cilia being damaged or destroyed. Although it is not clearly visible whether it's damaged or destroyed, since the question refers to describe the damage caused by B. pertussis, you can mention that the cilia being damaged just by looking at this certain area. You could also mention the presence of scar tissue. And for the explanation part, how does this affect the functioning of the epithelial tissue of the trachea? Well, if the cilia are damaged or destroyed, it affects the coordinated or synchronous movement of cilia. In other words, the cilia are paralyzed. If the cilia are paralyzed, then there is no synchronous movement. That is, the cilia being able to move from left to right. Since the cilia are not able to move in a coordinated manner, the mucus will not be moved effectively. Hence, it will cause the mucus to accumulate. Therefore, you could also mention the bacteria being trapped in the mucus and accumulating in the airways. And that is how it affects the functioning of the epithelial tissue of the trachea. Part B. Goblet cells produce mucus. Name one other structure in the gas exchange system that also produces mucus. The two main structures of the gas exchange system that you must know that produces mucus are the goblet cells as well as the mucus glands. Since the goblet cells is already mentioned in the question, the other structure would be the mucus gland. Part C. Suggest how whooping cough is transmitted. The question already mentions that it is transmitted from one person to another, similar to the disease TB. Therefore, since TB is an aerosol or droplet infection, this one also must be an aerosol or a droplet infection, as it is an airborne disease. Therefore, how is it transmitted? Well, when an infected person coughs or sneezes into the air, droplets will be produced that contain the specific pathogen, and these droplets will continue to remain in the air. Then when an uninfected person inhales the droplets, it leads to transmission of whooping cough, and those key points will land you 2 marks. Part D. The presence of P22 stimulates the production of mucin, a gel-like glycoprotein that is the main component of mucus. The mucin produced by the cell is packaged into vesicles ready for exocytosis. The first stage in the production of mucin involves transcription of the gene MUC5AC. Outline the stages occurring in transcription. Now the process of transcription occurs in the nucleus, and the final product is known as the mRNA molecule. Therefore, when you're referring to the process of transcription, you should mention that the DNA unwinds or uncoils, or you could also mention the DNA being unzipped. When the DNA uncoils or unwinds, it will form 2 strands. One strand is known as a sense strand, and the other strand is known as the antisense strand. And what happens when the DNA unwinds? Well, when the DNA unwinds, hydrogen bonds holding the 2 strands will break. Once the hydrogen bonds break, 2 strands will be formed, and as I mentioned, those 2 strands are known as the sense and antisense strand. Out of those 2 strands, one strand will act as a template for the production of a mRNA molecule, and that one strand which acts as a template is known as the antisense strand. In the process of transcription, one key enzyme that is involved and you must know is the RNA polymerase. It is not DNA polymerase, remember, because if the product being formed is an mRNA molecule, RNA polymerase is involved. DNA polymerase is only involved in DNA replication, not in producing mRNA. Therefore, what does the RNA polymerase do? The RNA polymerase, well, it forms the sugar phosphate backbone, as phosphodiester bonds form between the nucleotides that are brought and added to form the mRNA molecule. As you can see over here, free nucleotides are brought and added that are complementary in nature to the template strand. What is the complementary base pairing rule? It is where A binds to U and C binds to G, and using that complementary base pairing rule, free nucleotides will be added and it will form a sugar phosphate backbone. How does the sugar phosphate backbone form? Well, phosphodiester bonds are formed between the adjacent nucleotides. Finally, a mRNA molecule is produced, and that is how the process of transcription takes place. Part 2. Following translation, the polypeptide formed is modified by the addition of many short chains of monosaccharides in a process called glycosylation. Suggest where glycosylation occurs in the cell and explain why mucin is packaged into vesicles. Over here, the polypeptide formed is modified. And where does polypeptide modification or protein modification occur? Protein modification takes place in the Golgi body or the Golgi apparatus. Therefore, suggest where glycosylation occurs would be in the Golgi body. Then explain why mucin is packaged into vesicles. Well, mucin is packaged into vesicles in order to transport it towards the cell surface membrane. You could also refer to the bulk transport across the cell surface membrane, and you could mention the mucin acting outside the cell. Therefore, it needs to be released from the cell by using a process known as exocytosis in the form of vesicles. Those key points would land you 2 marks. Again, mucin is packaged into vesicles in order to transport it towards the cell surface membrane, because if you recall the process of exocytosis, vesicles they bind to the cell surface membrane. In order to transport it towards the cell surface membrane, vesicles are involved. Therefore, it is packaged into vesicles. Part E. Overproduction of mucus is one of the symptoms of chronic obstructive pulmonary disease, COPD. Describe the signs and symptoms that enable diagnosis of COPD. Possible signs and symptoms. If you recall the chapter involving COPD, it would be difficulty in breathing or shortness of breath, a persistent cough. You could also mention wheezing, difficulty to exercise due to shortness of breath again, feeling weak or feeling tired. Could also mention weight loss. Do not state the production of mucus as it is already given in the question. Hence, writing 4 possible signs and symptoms out of the ones given would leave you with 4 marks. Question 3. Outside the body, red blood cells can be maintained in an intact state by keeping the cells in a 0.9% solution of sodium chloride. This is known as a normal saline solution. Figure 3.1 shows intact red blood cells. Part A. Explain why red blood cells can be maintained in an intact state by keeping them in a normal saline solution. If the question mentions intact state, then you should mention that the water potential both inside and outside the cell are kept equal. There is no water potential gradient set up. If there is no water potential gradient set up, that means no net movement of water occurs or no osmosis occurs as a result. Therefore, in order to get 2 marks, you should mention that the water potential inside the cell and outside the cell are equal, hence leading to no water potential gradient being set up. As a result, no osmosis occurs. Part B. In the blood, red blood cells are suspended in plasma. The main component of blood plasma is water. Suggest one other component of blood plasma that could enter red blood cells and describe how it would cross the cell surface membrane. Possible components are oxygen, carbon dioxide, as well as glucose. The best possible answer to mention, since it refers to red blood cells, are gases such as oxygen and carbon dioxide. You could mention the gases being able to cross the cell surface membrane by diffusion down a concentration gradient because they are small molecules and gases are able to pass through the cell surface membrane. Hence, for gases such as oxygen and carbon dioxide, do mention diffusion down a concentration gradient through the partially permeable membrane. If you mention glucose, then you should talk about the facilitated diffusion process because glucose is too large to pass directly through the membrane, and you should mention facilitated diffusion requires transport proteins as well as it follows down a concentration gradient. Figure 3.2 shows red blood cells within a capillary. The capillary shown in Figure 3.2 allows rapid exchange of substances between the blood, tissue fluid and body cells. Part C. The actual diameter of the lumen of the capillary at the point X to Y in Figure 3.2 is 9.5 micrometers. Calculate the magnification of the image shown in Figure 3.2. Show your working. Magnification is nothing but image size upon actual size, and the actual size is already given, that is 9.5 micrometers. I have taken the image size as 9.5 millimeters, which is 9500 micrometers. It could vary according to the screen size you are using as well as the size of the given image you have. My measurement is 9.5 millimeters, which is 9500 micrometers, and once I substitute it into the magnification formula, I get the answer as 1000 because magnification is image size upon actual size. Part D. With reference to Figure 3.2, explain one feature that enables the surrounding body cells to receive an adequate supply of oxygen from the blood supplied by the capillary. The question mentions with reference to Figure 3.2. Therefore, if you look at Figure 3.2, you can see that the capillary is 1 cell thick, as well as red blood cells are very close to the body cells. Therefore, the features are red blood cells are close to body cells or capillary is 1 cell thick. The explanation that enables the surrounding body cells to receive an adequate supply of oxygen would be mentioning of short diffusion distances for oxygen to diffuse. The features are either of the two, and the explanation is short diffusion distance for oxygen to diffuse. Part E. Some areas of the brain known as blood-brain barriers have a type of capillary that is relatively impermeable to substances. Suggest one way in which the structure of a capillary in the blood-brain barrier differs from the structure of the capillary shown in Figure 3.2. Well, you could mention that there are no pores present in the capillary wall because normally Most of the capillaries have pores in their capillary wall. However, if the capillary is relatively impermeable to substances, then you could mention that there are no pores present or very few pores present. Question 4. Many microorganisms can digest cellulose by using a group of enzymes collectively known as cellulases. Cellobiose is a disaccharide produced during cellulose digestion. The cellulase known as beta-glucosidase completes the digestion of cellulose by hydrolyzing the cellobiose molecule to produce 2 beta-glucose molecules. Draw the ring structure of one beta-glucose molecule in the space provided. This is how the beta-glucose molecule looks like. The OH group lying on the first carbon atom, that is after the oxygen atom present in this ring structure, should lie above the plane. As you can see over here, my OH group lies above the plane, and it should be a hexose ring because glucose molecules, they are hexoses which has 6 carbon atoms present, and the carbons are not labeled over here. However, if you count in this given ring structure, you would get 6, and in order to draw the ring structure of one beta glucose molecule, you simply have to remember by referring to your biology textbook. Just study the key positions of the OH groups as well as the hydrogen atoms, as well as where CH2OH is placed. Part B. Beta-glucosidase was extracted from 2 different bacteria. Figure 4.1 shows the results of an investigation into the effect of temperature between 0 and 100 degrees Celsius on the activity of each enzyme. L represents the lowest temperature at which activity of each enzyme was detected. H represents the highest temperature at which activity of each enzyme was detected. As you can see, the graph is given over here, and the key is enzyme A represented as crosses, enzyme T represented as kind of circles. The first part, with reference to figure 4.1, describe the differences in the results for the 2 enzymes A and T. If you look carefully at this given graph, the optimum temperature of enzyme A is lower than the optimum temperature of enzyme T. As you can see, the optimum temperature of enzyme A is around 40 degrees Celsius, and for enzyme T it is around 85 degrees Celsius. Therefore, if we go back to the question, the optimum temperature of T is higher than that of A, or you can mention the reverse argument, that is, the optimum temperature of A is less than that of T. For T, the optimum temperature is around 85 degrees Celsius, and for A it is around 40 degrees Celsius. That is another key point, because the question refers to describe the differences, and for questions where it mentions describe, you should mention values, because values are a key part of this question. Then, after the optimum temperature, T has an initial steeper decrease than A. If you look carefully, for enzyme T, it has a steeper decline than at enzyme A. And A has a higher activity at lower temperatures, whereas T has higher activity at higher temperatures, because enzyme T is relatively more heat stable as it works at higher temperature ranges. A has a lower L than T, whereas T has a higher H than A. L represents the lowest temperature and H represents the highest temperature. And yes, L of enzyme A is lower than that of T, and H of enzyme T is higher than that of enzyme A. And once again, since it is a describe question, you should mention values. Part 2. Both enzyme A and enzyme T act on cellobiose. They have a similar but not identical primary structure. Suggest how similarities and differences in the primary structure of the 2 enzymes could help to explain the results obtained in the investigation. It is better if you could refer to how the primary structure is important. Well, the primary structure is nothing but the sequence of amino acids, and it determines the tertiary structure overall. Therefore, if you are looking at the similarities, you could mention they both have a similar active site since they both act on the same substrate or on cellobiose. Then you could mention the active site being complementary in shape to the specific substrate or cellobiose. as both enzyme A and enzyme T, they act on cellobiose, and in order to bind with cellobiose, they should have an active site that is complementary in shape. The differences are the difference in R group interaction, such as the bonds holding them. You could also mention why T is more stable at higher temperatures. It could be due to more bonds being present holding the structure together. Then you could mention the difference in type of bonds, such as hydrogen bonds, ionic bonds, disulfide bonds. This is a suggestion question, so you can write any key point that you feel is important to this question or is relevant. Question 5. Natural immunity and artificial immunity can both be acquired in a passive or in an active manner. Table 5.1 shows information about immunity acquired by two individuals P and Q. Complete Table 5.1. Description of event. Individual P is injected with a live weakened disease-causing organism. Outcome is person does not become ill from the disease and has long-lasting protection from the disease. Production of memory cells, yes or no? Well, this event is of a vaccination, and all vaccination events are artificial in nature. Therefore, it should consist of the word artificial in its immunity, and the outcome is the person does not become ill and has a long-lasting protection. That is active immunity, because only active immunity would lead to long-lasting protection and the person not being ill from the same disease. And active immunity is concerned with memory cells being produced. Therefore, for production of memory cells, you should put yes. Type of immunity would be artificial active immunity. Why artificial? Because it is a vaccination. Why active? Because the person does not become ill from the disease and has long-lasting protection. Let's look at the second event. Individual Q is injected with antibody against a specific disease-causing organism. Individual Q does not become ill from the disease but is ill with the disease a year later. If the question mentions antibody injection, it is always artificial and passive. Therefore, the answer is artificial passive immunity. Why artificial? Because antibody injection is artificial. Why passive? Because the person becomes ill with the disease a year later. If not now, the person will become ill after some years. Therefore, it is not active immunity. And since it is passive immunity, there will be no memory cells being produced, because memory cells are only produced in active immunity. Therefore, there should be no for production of memory cells, and the type of immunity is artificial passive immunity. Figure 5.1 is a light micrograph of a sample of blood. Cell X is a phagocyte. State the origin of the blood cell labeled X. All white blood cells are produced in the bone marrow, and phagocyte is a white blood cell. Therefore, it is produced in the bone marrow. Hence, the origin of the blood cell labeled X is bone marrow. What is this cell X? This cell X is a neutrophil because it has a lobed nucleus. Part C. Phagocytes play an important role when an immune response is initiated against cancerous tumor cells. Suggest how phagocytes can recognize the difference between healthy body cells and cancerous tumor cells. Healthy body cells have self-antigens, and cancerous tumor cells have non-self antigens. All cells will have antigens on its surface, and our body cells have self-antigens and are not recognized as foreign, whereas those that have non-self antigens are recognized as foreign substances. And phagocytes, they have the ability to distinguish between non-self and self antigens and will bind only to those recognized as non-self. How do phagocytes recognize those as non-self? Well, they have cell surface receptors for non-self antigens. Hence, in order to recognize the difference between healthy body cells and cancerous tumor cells, you should mention the phagocytes have cell surface receptors for non-self antigens, as well as the healthy body cells having self antigens, whereas cancerous tumor cells having non-self antigens. Part 2. Outline briefly how a tumor forms. Tumor is formed as a result of mutation. That is a key point. Then, what happens in the mutation? Well, a proto-oncogene changes to an oncogene. What does that lead to? Well, it leads to uncontrollable cell division, thereby forming a mass of cells. Because tumor is nothing but a mass of cells, and how does it form? It forms when there is uncontrollable cell division or multiple cell divisions. No programmed cell that occurs as a result because the cell keeps on dividing continuously. And these mass of cells that form a tumor do not respond to signals from other cells at all. And that is how a tumor is formed. The next question is regarding a topic that is not relevant in your current syllabus. Therefore, I will not be going through it. Hence, this is the end of this paper. If you found this video useful, do leave a like, comment, and if you haven't already, do subscribe. Thank you for watching and stay tuned for more.